Welcome to this edition of Clinical Clips, an accredited continuing education activity. This brief expert video will spotlight the daily hot topics from the conference.
Hello, my name is Dr. Joseph Mikhael. I am a professor at the Translational Genomics Research Institute and the Chief Medical Officer of the International Myeloma Foundation. It's a pleasure to share with you today in this clinical clip series about relapsed and refractory multiple myeloma that was presented at the annual American Society of Hematology meeting in Atlanta, Georgia. So I'm going to share with you briefly 8 key abstracts that were presented. And for each of them, the plan is really just to give you the key highlights as to what it's going to mean for our practice now and in the future. The first of these studies was the well-known Bellini study. This was a phase 3 trial comparing bortezomib and dexamethasone to adding venetoclax to bortezomib and dexamethasone. This study became well known because although it met its endpoint of improving progression-free survival in the total cohort, unfortunately, there was an increased mortality in patients who had also received the venetoclax. But this report was giving us an update on that, but also noted that in that smaller proportion of patients that have translocation 1114, and we know in these people they have an upregulation of the BCL2 pathway, that actually those patients can do quite well with this regimen with venetoclax involved. So I think the take-home message here is that we should not be using venetoclax generally in myeloma, but it can be used in patients with translocation 1114 with what appears to be improved progression-free survival and a trend towards improved overall survival. So more of that to come. It's not yet FDA indicated in that context, but I think it's very important to understand the different biology of patients that have translocation 1114. The second study I want to emphasize was about a novel agent called iberdomide, used to be called CC220. This is really part of a new class of drugs called CELMoDs that are, if you will, kind of cousins to the immunomodulatory drugs of thalidomide, lenalidomide, and pomalidomide. And so iberdomide has really cut its teeth in patients who are truly pomalidomide resistant. And at this update, we heard from Dr. Loniel how this drug can be very effective in very heavily relapsed patients with a response rate of about 26%, including the majority of those patients that were pomalidomide resistant. The other attractive feature to this drug is that it's a pill, and it seems very well tolerated, and perhaps indeed even better tolerated than lenalidomide and pomalidomide. So despite all of the new things we're going to be hearing about immunotherapies, having a simple and biological pill drug that our patients can take can be of tremendous value to them and will likely influence the field going forward. Well, another area, and the third abstract we're going to talk about is the first of the what we may think of as the bispecific agents. So these are agents that have, if you will, 2 arms, one that will hook onto the tumor and one that will engage a T cell. In the future, we're going to hear about other cells that can be engaged, like natural killers. But for this ASH, we're focusing on the T cells. The first of these agents was cevostamab. And what was interesting about cevostamab is that although we've been interested in BCMA-targeted bispecifics, this has a different target of FCRH5. I know these can be a little bit confusing, remembering all these letters. They all look like they're license plates, but FCRH5 is the target for cevostamab that engages T cells after it has locked itself on, as it were, to the FCRH5 molecule, which is really quite, or antigen, which is really quite prolific in multiple myeloma. So this was still early from a phase 1 study. There was some cytokine release syndrome, but thankfully, as we're seeing in these bispecifics, we're seeing very little, if any, grade 3 cytokine release syndrome, which is really important for the community doc ultimately, because that's obviously requires much more intense management than grade 1 and grade 2 cytokine release syndrome. So it was very fascinating for us to think about being able to use a drug like cevostamab because it does not target BCMA, as many other targets do now. Other drugs use that target. And so that may have a particular interest in attacking, if you will, the myeloma in a different way. Well, on the subject of different targets, abstract number 4 for us today is a drug called talquetamab. And I know it can be a little bit difficult to capture all these names. And it was in what would they call the Monumental, or TAL, Monumental 1 clinical trial. So this is also yet a different target than we're accustomed to. So cevostamab was FCRH5, and here talquetamab actually has a different target of GPRC5D. GPRC5D. And so GPRC5D is also something quite heavily expressed in myeloma cells and an attractive target. And this clinical trial, again, it's another bispecific agent that attaches this time to GPRC5D and engages T cells, showed a lot of single agent activity, was generally well tolerated. The cytokine release syndrome was tolerated. It does have a little bit of a different side effect profile than what we've been seeing before because of some GPRC5D expression on skin and hair cells. And so there are many patients that did experience some of this toxicity on their nails in particular, or in their hair with thinning of the hair, or skin changes, or effect on their nails, and indeed their taste. Now, thankfully, this does seem to be reversible in the overwhelming majority of patients, and generally mild, but something that we're not as accustomed to obtaining and are seeing in our patients. And so that's something to remember as we go forward with talquetamab. And then yet a third bispecific is abstract number 5, the one that you may have heard about before, because I'd suggest that it's the one that's the furthest advanced, and it may be even possible that it will go towards FDA approval in 2022. Of all of the bispecifics we've discussed, this is the one that's the furthest along, and it is called teclistamab, and this was in the Majestic study. You can see that a lot of this has to do with the name of the study itself. And the Majestic study was presented by Dr. Philippe Moreau. And now we have over 150 patients who have been treated with this agent. And again, we're seeing, as we saw with cevostamab and to a certain extent talquetamab, we're seeing literally a doubling of response rates of what we've historically seen in this heavily pretreated population. So in the single agents we've used in the past, we saw 25, maybe up to 30% response rates. Now we're seeing this, 50 to 70% response rate range in all of these bispecific agents. So really encouraging to see, especially in patients who, most of these patients are truly triple class refractory and indeed have been treated with just about everything available to them. So the teclistamab study was presented. Again, there was very, very limited grade 3 cytokine release syndrome, mostly manageable. There were the usual hematological side effects that we see in adverse events with cytopenias, mostly manageable. But again, although all of these studies are still admitting patients for their first dose, if not first couple of doses, we can see with time as we manage this better, this may be something ultimately that we could give entirely as an outpatient. That, of course, would be the goal in the long term. If we could do that for myeloma patients, I think it would make a difference. So keep an eye on teclistamab, because it may be the first BCMA-directed bispecific antibody, or the only bispecific antibody, or the next bispecific antibody that we'll see approved. And it has its BCMA target. So to quickly review that, cevostamab, FCRH5, talquetamab, GPRC5D, and teclistamab is targeting the BCMA, or the B-cell maturation antigen. Well, let's shift our focus for a minute from these bispecifics to the CAR Ts. We already have one CAR T available to us in clinical practice already in the form of ide-cel, and we may have another one early in 2022. But I'm going to share with you a couple of CAR T studies, the first of which was really building on work we've already seen with a similar product to the ide-cel product that's already available to us. But here it's the BB21217 as opposed to 2121, which ultimately became ide-cel. And the concept here was some biological engineering in this to be able to engage the T cells for longer. And so this was presented in the CRB402 study. This is, again, a BCMA-directed CAR T cell therapy. And again, we have been so impressed If we had great responses in the sort of 25 to 30% range with our single agents in the past, bispecifics are doubling that. We're starting to see that CAR T-cell therapies may even be tripling that. We saw that with ide-cel, we were over 70, if not in some cases over 80% response rate. And so although the numbers are small and preliminary, we saw about a 70% response rate in this early study. We expect the number to go higher as the full doses are being given. But really what we're looking for in this 21-21-7 study is, can it last for longer? So we saw in ide-cel that the median progression-free survival was about 9 months, and we're hoping that it may be exceeded as we biologically work on these molecules so that they can engage T cells more effectively and for longer. And that then brings me to our second last abstract, abstract number 7, which was highly anticipated, maybe the most anticipated abstract in the relapse setting at this meeting's ASH, which was the CARTITUDE study. So the CARTITUDE study is cilta-cel. So it is a different product than ide-cel that is being evaluated in patients with heavily pretreated multiple myeloma. And it was really quite remarkable because now we have about a year follow-up, a median of a little over 12 months follow-up, that there was a 98% response rate. I mean, literally a sort of a jaw-dropping kind of number. There was, of course, cytokine release syndrome, but the overwhelming majority of it was grade 1 and grade 2. And at 2 years, the median progression-free survival has not been met. So it does really look like we're going to exceed a PFS of 2 years. And these are patients that, by the way, we know historically haven't had a survival of much more than 6 to 9 months. So really quite remarkable to see this. In the 2 patients who hadn't responded, even there was some question as to the degree of their progression. So this is really quite remarkable. There were, in the earlier days, some neurological potential concerns about this drug and all of the CAR-Ts that we need to be monitoring closely, but there were no new neurological events here, and most of them seem to be very well managed. So that's very exciting, and that may well be the next available agent to us in myeloma as this is being evaluated by the FDA early in 2022. Well, I'm going to close off the abstracts with an important abstract, not so much a new molecule. We've talked about unique molecules like venetoclax. We've talked about unique molecules like iberdomide. We've looked at multiple bispecific agents and CAR-Ts. But this last study that I'm going to mention, it was one of many studies that are looking at real-world data and real-world evidence. And I think this is so important. Clinical trials are critical. Absolutely, we need to do them. But we need to see what's happening in the real world. And this real-world study wanted to look at what happens in patients who become triple-class refractory after they've seen proteasome inhibitors, immunomodulatory drugs, and monoclonal antibodies. What are their outcomes? And sadly, it really did demonstrate to us, and this was done from the CONNECT multiple myeloma disease registry, that these patients often aren't able to receive any treatment, have very poor survival outcomes, and indeed have a tremendous burden on their quality of life. And it's a reminder to us that although we're making all these great strides, we still have a very challenging disease to treat. And hopefully, as these bispecifics and as these CAR-Ts become more available and effective and hopefully less toxic, then this data that we have pointing to us to the severity of myeloma when someone becomes triple-class refractory can be overcome. So really an extraordinary meeting for all of hematology, an in-person meeting, which was very exciting to be a part of, but really tremendous data coming out in the sphere of multiple myeloma that I think is going to quite significantly change the way we treat myeloma in the not-so-distant future with novel molecules, bispecific agents, and indeed CAR-T cells. Well, thank you so much for joining me today in the ASH clinical clip on relapsed and refractory multiple myeloma. Make sure you view the other 2 clips that are being presented from ASH 2021. One of them is on CAR-T cell therapy and the other on low-risk MDS. Thanks again for joining us today.
We hope you found this activity useful and educational. To receive continuing education credit and to download your printable certificate, please return to the activity webpage and click on the Claim Credit button associated with today's clinical clip.